going to see about seeds. Seed formation. Seeds develop along with the fruit and reaches full size and maturity when the fruit ripens. The seed develops within the fruit by two distinct stages, that is, maturation stage, during which the seed loses water but continues to grow. Ripening stage result an increase in seed moisture content. Most of the seeds accumulate food materials during both of this stage. Seeds should only be extracted from ripened fruit. Seeds from immature fruit may not germinate under favorable condition, also loses its viability faster. The pollen grain adheres to the stigma, which contain two cells, a generative cell and a tube cell. The pollen tube cell grows into the style. The generative cell travels inside the pollen tube. It divides to form two sperm. The pollen tube penetrates an opening in the ovule called a micropyle. One of the sperm fertilizes the egg to form the diploid zygote. The other sperm fertilizes two polar nuclei to form the triploid endosperm, which will become a food source for the growing embryo. The zygote grows repeatedly by mitosis to form embryo. Embryo consists of plumule, radicle, cotyledons. The endosperm nucleus divide repeatedly to form the endosperm in endospermic cells. Endospermous monocotyledons, example is onion. Endospermous dicotyledons, example coffee. In non-endospermic seeds, the endosperm is used up in the early stages of seed development, so the food is stored in the cotyledons. Non-endospermous monocotyledons, example is orchid. Non-endospermous dicotyledons, example mango, jackfruit and pea. Embryo, whole dormant immature plant inside the seed. Radicle, part of the embryo which will become the root at germination. Plumule, part of the plant which will become shoot at germination. Cotyledon, this is the leaf which act as food reserve or absorb nutrients during germination. Testa, protective coating of the seed formed from the integuments of ovule. Seed storage. Seeds are stored at relatively dry condition at low temperature. Seeds like citrus, rubber, cocoa, mango should be sown immediately as seeds are short-lived. Seeds can retain its viability by storing at low moisture content, reduced temperature, which slows respiration rate at other metabolic activities without damaging the embryo. Based on storage, two types of seeds are there. Orthodox seeds are long-lived seeds and can be successfully dried to moisture content as low as 5% without injury and are able to tolerate freezing. Example, lantana camara, guava, cashew, citrus. Recalcitrant seeds are remarkably short-lived, which cannot be dried to moisture content below 20 to 30% without injury and are unable to tolerate freezing. Example, avocado, cacao, coconut, jackfruit, lychee, mango, rubber. Seed germination. Cell metabolism increases, the embryo resumes activity growth, seed coat ruptures and seedling emerges. The water softens the seed coat. Enzymes become active and digest fats, proteins, carbohydrates in storage tissues. These compounds are transferred to embryonic axis to be used for growth of plant. The seedling Stem is divided into two sections. The section below the cotyledons as hypocotyle, the section above the cotyledon as epicotyle. Epicotyle and hypocotyle difference. Epicotyle. Area of embryo axis between the plumule and cotyledonary node. In hypogeal germination, epicotyles elongate and seeds remain underground. Hypocotyle. Area of embryo axis between the radicle and the cotyledonary node. Its apigeal germination, hypocotyles elongate and push the seed out of the soil. Seed viability and longevity. The capability of the seed to germinate and produce a normal seedling for some specific time is called seed viability. Factors affecting seed viability. Pre-harvest factors: seed coat, pollination, mechanical damage, microflora, time. Post-harvest factors: moisture, temperature, relative humidity, light, chemicals, insects and mites. Seed viability test. Germination test and tetrazolium test. Tetrazolium test. Seed hydration. It is done by soaking seed in water for a specific period of time. This is done to activate hydrolytic enzyme and stimulate respiration. Cutting or rupturing of seed. This is done to allow the penetration of triphenyl tetrazolium chloride solution into the internal tissues. Staining of seeds. It is done by soaking seeds in triphenyl tetrazolium chloride solution for a specific period of time to allow staining of viable tissue in the seed. Tetrazolium chloride is used at 0.1 or 1.0% solution at 30 to 35 degrees Celsius temperature for 24 to 48 hours at pH of 6 to 7. Viable seeds take red color, non-viable seeds remain colorless. Technique of seed propagation. Pre-treatment of seeds. Special treatments are necessary for seeds of many plants to germinate even when conditions appear favorable. Mechanical scarification. It includes breaking or scratching the seed coat mechanically to modify the hard or impervious seed coat. Soaking in water. Generally, seeds will be soaked in hot water for a few seconds and then soaked for 24 to 48 hours in cold water, which makes the seed coat to soften. Acid treatment. Soaking the seeds for a few minutes in concentrated hydrochloric acid or sulfuric acid modifies the hard impermeable seed covering. Cold stratification. It consists of placing the seeds in a moist medium of sand, peat, holding slightly above freezing temperature. Chemical treatment. Soaking in potassium nitrate at 0.2%, gibberellic acid 200 to 500 ppm or thiourea at 0.2% solution prior to sowing stimulates germination.